Welcome to Part 4 of this series of presentations that describe the mammalian stress mechanism postulated by Hans Selye. This slideshow will present pertinent attributes of the vascular endothelium, which is the focus of all stress mechanism activity. The vascular endothelium is a diaphanous layer of cells, one cell thick, that lines the inner surface of all arteries, arterioles, veins, and venules. It insulates flowing blood from extravascular tissues. It produces several stress mechanism substances and enzymes, including von Willebrand factor, tissue plasminogen activator, nitric oxide, fibronectin, vitronectin, thrombin-activated fibrinolysis inhibitor, protein C, and stoichiometric AT3. The vascular endothelium is the sole constituent of capillary walls, which are only one cell thick. Capillary surface area is vastly greater than that of all other vessels combined. Capillary pressure, flow, and turbulence are negligible compared to larger vessels. Capillaries have no muscular elements that enable vasoconstriction or capillary hemostasis. The green cells on the left portray healthy vascular endothelium that insulates flowing blood from extravascular tissues. The gray cells in the center represent gap formation between endothelial cells induced by thrombin, radiation, and toxic chemicals, which increases contact between blood and extravascular tissues. The flesh-colored cells on the right portray disruption of the vascular endothelium by trauma and surgery, which directly exposes blood enzymes to tissue factor. Factor VII in flowing blood slowly but continuously penetrates the healthy vascular endothelium to enter extravascular tissues, where it is activated by tissue factor. In the absence of factor X, it generates small amounts of thrombin that energize tissue maintenance. Tissue factor slowly leaks into flowing blood, where it activates factor VII and generates small amounts of thrombin that activate factors 8, 9, and 10. The vascular endothelium produces and releases stoichiometric AT3, fluctuating protein C, and tissue factor pathway inhibitor that continuously quench and control factor VII activity. This normally controls thrombin generation and flowing blood within a narrow range, but these inhibiting mechanisms can be overwhelmed by stress-induced enzyme hyperactivity and positive feedback that produces the manifestations of disease. The gigantic size of factor VIII prevents it from penetrating the intact vascular endothelium or inflammatory gaps in capillary walls, or even the viscoelastic clot of its own making, so that it becomes fully active only when the vascular endothelium is completely disrupted by trauma or surgery. This explains why radiation, toxic chemicals, and sepsis do not induce clot formation. Factor IX amplifies factor VIII activity but does not interact with the other blood enzymes, so its ability to penetrate the vascular endothelium is irrelevant. Factor X cannot penetrate the intact vascular endothelium, but it is small enough to pass through inflammatory gaps in capillary walls induced by sepsis, toxic chemicals, and radiation. It also penetrates the viscoelastic clot so that it interacts with factor VII and tissue factor in damaged tissues and elevates thrombin generation to levels that sustain tissue repair. This interaction of factors VII and X with tissue factor is the engine of tissue repair and embryological development. Radiation, toxic chemicals, and sepsis increase the permeability of the vascular endothelium and allow factor X as well as factor VII to penetrate the capillary walls, which elevates thrombin generation to levels that sustain tissue repair. Factor VIII cannot penetrate the intact vascular endothelium on account of its gigantic size, which explains why clot formation is absent in these conditions. Radiation, toxic chemicals, and sepsis also increase the leakage of tissue factor into flowing blood, which exaggerates thrombin generation in flowing blood, causing harmful systemic inflammatory effects and positive feedback. Surgery disrupts the vascular endothelium and releases tissue factor into flowing blood, which abnormally exaggerates factor VII activity, overwhelms inhibitory mechanisms, and induces positive feedback that reaches peak intensity 48 hours later. This explains why the manifestations of surgical stress occur distant from the time and location of surgery, and why they are related to the degree and duration of surgical tissue disruption. Major trauma causes similar consequences. Tissue disruption directly and simultaneously exposes tissue factor to all four blood enzymes. Tissue factor activates factor VII to generate small amounts of thrombin that initiates the interaction. Factor X elevates thrombin generation to replicate itself. Factor VIII further elevates thrombin generation to energize its conversion of factor X to factor XIII to generate insoluble fibrin. And factor IX exaggerates factor VIII activity. This powerful interaction enables coagulation that is limited to the area of tissue disruption because factor VIII cannot penetrate the viscoelastic clot of its own making. The viscoelastic clot stems blood loss, but its more important function is to regulate tissue repair. This will be discussed in detail in the next slideshow. The vascular endothelium is specialized to optimize function in different organs and tissues. The blood-brain barrier consists of endothelial cells with tight junctions that limit permeability. Compared to brain tissue, the endothelial cells of renal and hepatic tissues have loose junctions that optimize the function of these organs. The vascular endothelium has both sympathetic and parasympathetic innervation in vital organs such as brain, lung, and bowel. Electronic signals also travel from cell to cell in the vascular endothelium, so that direct autonomic innervation is not necessary to govern each and every cell. Sympathetic nervous system stimulation causes the vascular endothelium to release von Willebrand factor into flowing blood, which exaggerates factor VIII activity and increases insoluble fibrin generation in capillaries and flowing blood. This has widespread effects. Insoluble fibrin increases blood viscosity and inhibits tissue perfusion. The intense sympathetic nervous system stimulation activates capillary hemostasis and causes infarction. Parasympathetic nervous system activity induces the release of nitric oxide from the vascular endothelium. Nitric oxide binds to calcium, inhibits thrombin, accelerates insoluble fibrin disintegration, and promotes tissue perfusion and organ function. Epinephrine and insulin are hormones that extend autonomic effects to peripheral tissues, such as skeletal muscle, where direct autonomic innervation is lacking. Sympathetic nervous system activity stimulates the release of epinephrine from the adrenal glands, which releases von Willebrand factor from the vascular endothelium into flowing blood and increases insoluble fibrin generation. Parasympathetic nervous system stimulation releases insulin from the pancreas, which induces the release of nitric oxide from the vascular endothelium and disintegrates insoluble fibrin. Brain astrocytes release both tissue plasminogen activator and nitric oxide that disintegrate insoluble fibrin and preserve brain perfusion. Their activity prevents brain infarction when large amounts of epinephrine are administered during cardiopulmonary resuscitation. That is enough for one slideshow. The next slideshow will explain the tissue repair mechanism.